So let's move to the next speaker. Next speaker is Rosanna Olsen. She is from Rotman Research Institute, Baycrest Academy of Research and Education, Toronto, from Canada. The title of her presentation is a structure and functional division of the medial temporal lobe subregion, historical overview and practical guidance of neuroimaging. So please welcome Rosanna.
Hi, everybody. I'm really excited to be here, and I'm also really excited to be going in this position because I think you'll see that a lot of the content of my talk is going to be reflected and reminiscent of some of the other themes that you've heard from the other speakers. I also thought I'd make another sort of meta point, which is, you know, when you attend OHBM, especially if you do functional imaging of the medial temporal lobe or any other region of the brain, you might kind of take for granted the fact that you might think, well, we already know what the structures are. You know, we just need to map out the functions. The structures were discovered, you know, hundreds of years ago. But I think what's apparent from many of the talks that we've heard from today is that actually we're still learning quite a bit about the neuroanatomy, and that's an exciting time that we can be learning about both things. I have no disclosures. So let's talk about what we do know. And of course, much of this work has been established by, you know, extensive studies using tracer studies as well as electrophysiological studies, stimulation studies, as well as careful anatomical and histological studies of the different regions of the medial temporal lobe so that we can better understand these distinct subregions. So the medial temporal lobe we can think of as having kind of two major subdivisions. One is the hippocampus, and then the other part is the medial temporal lobe cortex. So the hippocampus itself also has subregions within, and those we usually think of as CA1, CA2, CA3, subiculum, and dentate gyrus. And then the medial temporal lobe cortex contains entorhinal cortex, perirenal cortex, and parahippocampal cortex. And in general, we think that information flows, at least on this diagram in a counterclockwise fashion, where information goes from the entorhinal cortex into the hippocampus via the dentate gyrus, up to the CA3, over to CA1, down to subiculum, and then out, subiculum being the major output structure. Of course, there's also direct connections between the entorhinal cortex and other subregions of the hippocampus itself. And like I mentioned before, we know a lot of what we know about the human MTL actually has been gleaned from studying both rodents as well as non-human animal primates, and some of the beautiful work that we've heard from the previous speakers, as well as from Menno Witter, for example, has shown that we know about the connections, again, through this sort of circuit from entorhinal cortex into the hippocampus and back out again. We also have come to appreciate that the regions, the different layers within the different cortical regions have specific projections into different layers of the hippocampal subfields. So it's much more complicated than the diagram on the left, which just sort of shows this one circuit, but then when you actually look at the layers, you can see that there's specific inputs from layers to specific layers in the hippocampus, and then from different layers of the hippocampus out to different layers of the MTL cortex. So very fine-grained structure. Now, just taking a step back and just making sure that we have a broad understanding here and kind of level setting in terms of the different directions at which we think that the hippocampus and medial temporal lobe are organized. So I've already showed you that trisynaptic pathway, or that loop. One other actually really important organizational scheme within the medial temporal lobe is just simply from anterior to posterior. So front of the brain, anterior, posterior is the back of the brain. And if we look at a coronal slice of the hippocampus, we're taking a section, you know, like this, and what we see is the hippocampus on both sides of the brain. We have two of them, just like the thalamus, and the medial part is towards the middle, and the lateral part is towards the outer part. And then we can also talk about the superior or dorsal part of the hippocampus versus the inferior ventral part. Now, again, I said that much of what we know about the organization of the hippocampus was based on what we know from animals. And you might be wondering about how you can convert sort of thinking about the functions of the anterior hippocampus and the posterior hippocampus compared to different regions. For example, in the rodent, who actually don't use those terms when they describe the different regions of the hippocampus. And in this 2014 paper by Brian Strange, he explains sort of the axis that we think of as anterior to posterior. When you go to the rodent, it's ventral to dorsal. So the ventral hippocampus in the rodent is highly connected to the amygdala, for example, and is thought to be involved in emotional behaviors. The dorsal hippocampus is thought to be involved more in spatial navigation behaviors in rodents, and we have sort of a similar organization in humans when we go from anterior to posterior. So this is what it looks like in a sagittal MRI, and many of you might be familiar with the kind of tripartite structure of the hippocampus being the head, the body, and the tail. And so as you go from anterior to posterior, you have the head and the most anterior part. The body and the tail often get grouped together for kind of convenience reasons. And many people, when you're doing functional MRI, at least standard resolution functional MRI, will group together the head of the hippocampus separately from the body and the tail. And there's been now many studies that have looked at the different functional contributions of head versus body and tail. Now, in that picture of that sagittal view of the T1, when I labeled head, body, and tail, it's ignoring, of course, the subfield structure that I explained at the very first part of my talk. So in the head of the hippocampus, you actually have subiculum and then CA1. And then as you move posterior from the head of the hippocampus backwards, you have CA2 and CA3, then the dentate gyrus, et cetera. Then in the body of the hippocampus, you have all the subfields all together. And then the tail of the hippocampus, you still have all of the subfields, but the tail of the hippocampus is very difficult to image in humans, and so we often group that region together when we're doing functional imaging, as shown in this slide. Okay, so many of us who study the hippocampus have this book on our, at least saved on our hard drive or sitting on our desk. It's by Duvernoy, and he does a beautiful job of explaining the subfields and different definitions of the hippocampus. And so I'll give you sort of a historical overview of how some of these areas are named. So hippocampus proper is usually, and again, the nomenclature will differ across neuroanatomy labs, but some neuroanatomists say that the hippocampus proper is CA1, CA2, CA3. The reason why CA4 is in italics is because not all neuroanatomists believe that CA4 is a separate subregion. So in Jacob's talk, he talked about the prefrontal cortex and all the different subdivisions that different neuroanatomists over the years have given to different regions. Same exists in the hippocampus, where not every neuroanatomist agrees that a distinct piece of tissue is a certain name. They disagree on the name. Then when you go to the hippocampal formation more broadly, that includes the dentate gyrus, the prosubiculum, again in italics, the subiculum, presubiculum, parasubiculum, and entorhinal area. The prosubiculum is an interesting area. It's right in between CA1 and subiculum, and some neuroanatomists claim that it's a very distinct area cytoarchitectonically. Others say that, no, it's just a transition region between CA1 and the subiculum. So maybe fMRI and maybe functional imaging can shed a little bit of light on this, because if we can see functional differences between CA1 and adjacent prosubiculum and subiculum, maybe we can argue that that is a distinct anatomical region. Now, one thing I wanted to point out, and Duvernoy also illustrates here, is that the neurons, when you look under a microscope in CA1, CA2, CA3, CA4, and DG, they look different. Why do they look different? Well, sometimes in some regions, the pyramidal cells are larger than others. They're also more densely packed than others. And those are the histological features that the neuroanatomists used when they defined these subregions. Dentate gyrus is the most different from all the other subregions. That's because it contains more, as I'll show you in the next slide. I don't think it has either very few or no pyramidal cells, and mostly consists of granule cells and basket cells. So that's why, again, if you look at staining of histological tissue, you can see that the dentate gyrus looks very, very different from the CA fields. Now, one thing that I wanted to talk about was, again, sort of the dimensions in which we think the hippocampus is organized. So I already told you about the subfields. I told you about anterior to posterior. I didn't talk about superficial versus deep. And so that's the third axis of organization that Zembrowski and Sprusten in this review paper have pointed out are important for understanding the functional subdivisions of the hippocampus. So it's the case that along that transverse axis, we know that function will vary along that transverse axis as you move from dentate gyrus to CA3 to CA2 to CA1 to subiculum, as well as you move from the outer part of the hippocampus to the deeper layers, and I'll show you some examples in a few slides of those different layers. Similarly, I already told you that along that dorsal to ventral axis, or anterior to posterior, if we're looking at a human, we see very different functional activations, for example, using fMRI. Also, the size of, for example, place cells differs as you move from anterior to posterior hippocampus. So we do think that there are major functional differences and structural differences as you move from dorsal to ventral. So last circuit diagram I'm going to show you, I promise. This is again just to point out that much of what we actually hypothesize is the function of the hippocampus is drawn from what we know about the anatomical organization, and in fact, specific hypotheses about the functions of, for example, the recurrent connections in CA3 have given rise to hypotheses about what kind of mnemonic function that area might play, as well as projections again throughout that loop. So you might be wondering, I've showed you some high resolution MRI, I've shown you some sort of schematics of the brain. Well, what does this circuit look like in the human? And for that, I'd like to point you to this beautiful work by Michael Zilles from 2016, where not only does he label on a high resolution MRI image showing you where the subfields are, but he's also showing where the white matter structures that connect the different regions, subregions of the medial temporal lobe, where those approximately are. And he's actually done, this research has come out of some really cool polarized light imaging that I'm going to show you in a second, where you can look in very, very high resolution to see these distinct pathways, not only along the transverse axis, but again along the longitudinal axis of the hippocampus, connecting regions like the CA3 and the CA1, et cetera. And that's one of the images from his beautiful 2016 paper, where you can not only see, one of the things that I wanted to really point out in this image is that you can see the beautiful layered structure of the hippocampus. And so some people, especially if you do kind of standard resolution MRI, you might just think of the hippocampus as one big blob, but actually it has these beautiful layers. And as you know, if you've read Duvernay, and the layers that contain the pyramidal cell neurons is called the stratum pyramidalis. Sorry, my Latin is not very good. And then you can see the stratum radiatum, stratum lacunosum, stratum moleculare, the hippocampal sulcus, et cetera. And so that layered structure is really important. But remember, I showed you on one of those other circuit diagrams, because the different layers of the medial temporal lobe cortex are projecting to different layers within the hippocampus. And so this is just a reminder, hippocampus is not a subcortical region as it's sometimes thought of in the neuroimaging world. It's actually a part of cortex or allocortex. Okay, so what about imaging the white matter in vivo? The previous study that I showed you by Michael Zilles was an ex vivo study. He's also done some in vivo imaging of the collateral, the Schaefer collateral, using DTI. And I also wanted to point out the work of my colleague Marshall Dalton, who has done some really beautiful work. I believe this is using Human Connectome Project DTI data, where he's looked at streamlines between the hippocampus, specifically the posterior hippocampus and V1, as well as projections between the hippocampus and TE, might be TF, ask Marshall afterwards, and showing that the endpoints for V1 projections are very specific to the tail of the hippocampus, whereas the TF regions are more distributed along the long axis. So really elegant work, and I think he's going to speak about it more in the Thursday symposium as well. And then in addition to streamlines using diffusion-weighted imaging, we can use regular kind of structural volumetric measures to assess the integrity of white matter structures within the medial temporal lobe. Of course, you need very high-resolution data for this, but my colleague Robert Amaral in 2016 published a paper where he not only looked at the gray matter regions of the medial temporal lobe, but also looked at white matter regions like the fornix, the fimbria, and the alveus, and segmented them to measure volume and to examine how those structures change with age and neurodegeneration. Okay, so Many of you might be interested in how to segment the MTL because you want to know, okay, what are the things that I need to know if I'm interested in trying to figure out what the difference is between the function of CA1 versus the dentate gyrus, for example. Okay, so what do you need to know? Well, first of all, you need to be able to localize where those structures are. Remember I said medial and lateral, so CA1 is on the lateral side of the hippocampus, as you can see there. DG is very medial. And the thing that separates them is this something called the dark band. And the dark band is made up of those layers that I described before that I'm going to abbreviate SLRM, or stratum, SRLM, stratum radiatum lacunosum moleculare. And this is a region of the brain that you can only visualize if you have very high resolution data, and it's visible in this coronal plane, and you have to be able to, again, be looking at voxels that are probably less than a millimeter. And I'll have another slide in a minute when I recommend what resolution to obtain to look at that. And again, Duvernoy has a very nice cartoon images of what those different layers are, what kind of neurons and what kind of structures are contained within the, I always like to go back and forth between these. This is a very high resolution postmortem MRI scan. I think it's 9.4 Tesla, so that you can see the exact structures. But even using in vivo MRI, you can get nice images of the hippocampus to be able to identify the SRLM. And what that allows you to do is it allows you to separate the CA1 laterally from the dentate gyrus medially. Okay, so how big are the hippocampal subfields? Jacob recommended using very high resolution functional imaging. I'm also going to recommend doing that because the hippocampus is very small. So from medial to lateral, the hippocampus is only about 1.5 centimeters big. Along its long axis, it's only about 3 to 4 centimeters big. So you obviously need very high resolution data in order to image that region. Now, if you want to look at subfields, of course, it's even smaller, and people who've done ex vivo as well as in vivo imaging of the hippocampus have provided measurements of these different the width of CA1, and it can be in some sections, and especially in an older adult, it can only be 1 millimeter thick. So if you're collecting 1 millimeter thick, 1 millimeter cubic voxels, then you're going to have a very hard time establishing the spatial location of that region. So what kind of data can you collect? Of course, there's many different kinds of images that you could collect to image the hippocampus. One of the really popular ones is to use T2 weighted coronal images. For example, ADNI does this where it has a very high resolution in-plane acquisition, 0.4 by 0.4 millimeters with a thicker through plane or slice thickness. Of course, you can, with higher fields, you can get maybe a little bit better in very thin slices, which is even better. But one of the things to point out is that you want to have an acquisition. If you're not doing whole brain acquisition, you could do something like a slab, like what Andy does. You can just cover the hippocampus itself, which is like the white box with the dashed lines. Or if you want to make sure you're scanning the perirenal cortex as well, then you need to extend that all the way forward to the temporal pole so that you can include the perirenal cortex as well. And I'm going to talk about the hippocampal subfields group in a minute, but just for your reference here, we give some recommendations on our website about that. So remember, I just told you that the width of CA1, especially in older individuals, can be quite thin. And if you're scanning at 1 millimeter isotropic, it is very difficult to be able to see those, have good enough resolution to measure those structures reliably. And so this is just a paper that we wrote a few years ago where we argued that you really need this high resolution in-plane scan. It doesn't necessarily have to be T2 weighted, but that's kind of how it was originally developed, but something that gives you the fine enough resolution so that you can see that dark band or the SLRM, as well as other features of the hippocampus like the dentations on the top of the hippocampus. You can see that that's really only visible when you have a resolution that's high enough to be able to visualize those important structures that we think might be meaningful. And we heard before, maybe recommending something on the order of 1 millimeter cubed. That's a really high resolution. It's hard to get, and it's hard to get whole brain. But for something like areas of the medial temporal lobe, and especially if you're interested in scanning layers of the medial temporal lobe, you're probably going to need something on the order of 1 millimeter isotropic or even smaller. If you don't care so much about the layers, but especially maybe you're more interested in the long axis organization, you can probably get away with something more like 2 millimeters isotropic. Okay, so this nicely also, I would like to talk about the variability of the anatomy of the hippocampus, which is fascinating to somebody like myself, and hopefully also interesting because we just learned about the different anatomy of the tertiary sulci and the prefrontal cortex. So one known variant of the hippocampus is within its shape in this, seen in this coronal plane. There is a variant called hippocampal inversion, which we think occurs between 5 and 20% of healthy individuals. It could also occur in higher rates in other diseases such as epilepsy. But you can imagine that if you're doing sort of standard resolution MRI and you have these different shaped hippocampi, that might cause some problems. The other thing that is really fascinating is that the sort of more global morphometry of the hippocampus can vary quite a bit from person to person. The hippocampal head is one area that really differs across people. So those dentations on the superior aspect of the hippocampus that I showed you before, people can have between 1 and 4 dentations in their hippocampus. And so that is highly variable across people. We don't actually know what the function is of those regions and whether, better memory, for example, could result in more dentations versus fewer. Those are all exciting open questions. But nonetheless, this is just to show that in this beautiful work by Robin DeFlores and David Barron, that the anterior hippocampus, or the head of the hippocampus, varies greatly in terms of its overall size and shape across individuals, and the tail of the hippocampus. So sometimes the tail curves more medially, sometimes it curves superiorly, and other times it curves inferiorly. So it's very complicated, and again, depending on the person's anatomy, they can differ, and they don't have to be the same on both sides. They can have one shape on one side and another shape on the other. Okay, another source of variability within the medial temporal lobe has to do with the blood supply. So when you look at large vessels in the hippocampus, or in the brain overall, like for example, the circle of Willis, almost everybody has the same number of arteries in their brain, and they connect in almost identical way. However, when you go to the smaller arteries, the branching arteries, that's where you start to see variability. So kind of similar to those primary versus tertiary sulci that we talked about before. In the hippocampus in particular, we see variability in the number of arteries that have branching arteries into the hippocampus. So by and large, most people will have branching arteries feeding their hippocampus from the posterior cerebral artery, the PCA. However, some individuals have what's called the dual supply. If you have a dual supply, you have extra blood supply to the anterior part of your hippocampus from the anterior choroidal artery, or the ACA. So this was first described, different patterns have been described for many years using postmortem data, but it was first described using MR angiography by my colleague Valentina Perosa. She looked in older adults, and she looked at individuals that had either a dual supply versus a single supply, and she showed that people with a dual supply have a larger anterior head of the hippocampus, and they do better on a California verbal learning test. So this is an exciting new direction that I'm exploring in my lab, where we're collecting. One thing I should say is that Valentina's work was at 7T, so in order to image the ACA especially, it's very difficult with using kind of standard 3 Tesla MRI. You can do it, but it's a little harder. So it's important to sort of validate some of these methods using 7T MRI. So I have a collaboration with University of College London with Martina Callahan and Emma Zilles, where we're scanning some of the same individuals at 7 Tesla and 3 Tesla and characterizing the blood supply to their hippocampus. And so one of my graduate students, as well as a graduate student working with Jean Chen, is working on manually delineating the PCA as well as the ACA in all these individuals. Now, just like the hippocampus, now when we come to the MTL cortex, it's even more variable than the hippocampus. So anytime you move away from sort of these older structures, I think, as you get to these sort of newer structures, more complicated structures, association cortices that are, are newer, they get a little more complicated. And so these are the kind of things that, for example, Ricardo and Sausti described in his seminal 1998 paper, where he described all of the different things that can vary across individuals in terms of their collateral sulcus, which is the next sulcus over from the hippocampus. So it's just lateral to the hippocampus. So the anterior extent of that collateral sulcus can differ across people. The depth of the sulcus can vary across people, as well as the collateral sulcus itself can be either bifurcated or interrupted. And my colleague Valerie Karl made some nice slides to demonstrate this. So she showed that the collateral sulcus in this individual is bifurcated. So the red area is the collateral sulcus, and so she shows in that most anterior slice they only have one collateral, and then in the slice more posterior to that, there's two collaterals. And then you can look in the axial view to see, oh, that's because there's one collateral sulcus that's coming in from the anterior part, then it jumps over and it starts again more lateral to that. And we would classify both of those sulci as part of the collateral sulcus and probably have both of them have perirhinal cortex neurons. But you can imagine how much of a headache this is for us as imagers in trying to understand the functions of these regions if they're so variable across people. Similarly, in other individuals, there's not just the bifurcation, but there's actual a disappearance completely of that sulcus. So the sulcus just shrinks down as you're moving from anterior to posterior and then reappears later, more posterior to that. And Prusner and colleagues have demonstrated that about 20% of participants, I would say maybe even more in my experience, have these variants. So now I'd just like to tell you a little bit about some of the harmonization work that myself and as well as many other labs have been working on in the past 10 years or so in trying to come up with a common reliable protocol that we can use to segment hippocampal subfields given all of these problems that I mentioned before. And one of the problems that we noticed in the field is that different labs were actually segmenting. I mentioned before that different neuroanatomists disagree, so might not be a surprise to you that actually different neuroanatomy labs disagree in terms of the number of subfields there are in the hippocampus and where those boundaries are across those regions. And so in 2015, we put out this paper where we had each lab take the same MRI, segment it using their own lab protocol, and we demonstrated that there was remarkable variability across the way that the definitions that people were using to place the boundaries between regions like CA1 and subiculum, for example. And maybe you think, well, what does it matter if the boundary between this lab and this lab is over by 2 millimeters versus another? But I think it can matter if we're trying to really know precisely what different subfields are doing in terms of different mnemonic functions, as well as how different subfields are changing as a function of age and disease. And so this figure kind of nicely demonstrates the variability across the field, where across all of these different studies, all of these studies found regions that were changing as a function of age. For example, the yellow regions were, the only difference between yellow and red is sort of the method, the type of MRI they used. But you can see there's not a whole lot of consistency across these labs in terms of which regions were found that were changing with age. And one other thing to point out is that, for example, my two colleagues, Renaud Lajoie and Laura Weiss, they really differed in terms of where they were placing that boundary between CA1 and subiculum. And so you can imagine that if they were using the same boundary, they might have found the same results. So that's why we are trying to form the harmonized protocol. Now, of course, there's many other differences across these studies, such as the composition of the subjects, so we can't say it was only due to boundary differences, but at least we can try to take away at least one source of variability. So just in the interest of time, I'm not going to be able to go through our entire harmonization procedure, but this is just to say that we worked with a number of neuroanatomists to try to better understand where the variability had come from across different atlases. Was it simply that they were using different staining methods, and that's why they had different rules in terms of labeling the different regions? Was it that the specimens that they used to publish the atlases, I just mentioned there's a lot of anatomical variability across individuals, maybe that's driving the differences. And what we found that was quite interesting to us is that even when they're using the same stains, looking at the same brains, they still nonetheless had different definitions of the subfields. And so we've worked with them, and they've been very generous with their time in terms of helping us with the protocol development. And I'm happy to report that the first part of our procedure is finished, so that we have a published protocol that's now available for download for the hippocampal body, and we're very close to finishing the hippocampal head, hippocampal tail, and now we're moving into the MTL cortex. And so if you want to download the protocol for the hippocampal body, you can visit our website. And this is, again, just to say that we're working on the MTL cortex. And again, as I mentioned before, there's a little bit of disagreement in the hippocampus, and then when you get into the entorhinal cortex, it gets even more different across neuroanatomists. The good news is entorhinal cortex, all of our neuroanatomists were very consistent with each other, but in fact, even just the definition of perirhinal cortex differs across neuroanatomists. So some people group 35 and 36, Brodmann area 35 and 36 together. Others keep them distinct, and Brodmann area 35 and Brodmann area 36 were also pretty variable across neuroanatomists, but we're taking all that into account when we're developing our MRI protocol. And I think I'm running out of time, so I'm going to skip this part. I also just wanted to give a little bit of a plug for another paper that the hippocampal subfield group work, which is about how to perform quality control on automated segmentation. So one of the things that you might be interested in is, how do I look to see if my segmentation worked? If I'm using automated segmentation, we gave some guidelines for that and recommendations for students who want to use this in their work. Finally, I'd just like to mention a couple of resources. In addition to the hippocampal subfield group website, you can also look at the YouTube channel by my colleague Laura Weiss. If you're new to the MTL and you want to learn more about MTL anatomy than I just told you, you can watch some of her videos on YouTube. They're really useful. There's also a really nice, beautiful animated video from Johns Hopkins University that shows the nice definitions of the different layers and the different subregions. So please check that out. I just want to say thanks.
So thank you very much. So we have time for taking a couple of questions. So if you have any question, please go to the microphone. Okay, please.
Hi, and thanks for the talk. I would like to hear your thoughts on preprocessing. So, because we keep using very aggressive nonlinear registration algorithms, and on top of these parcellations,
right?
Are we doing everything wrong?
Yeah, so in my lab, because I'm so interested in individual subfields, I actually typically don't collect, I'm not going to say never, but many of the experiments that I've run have actually not looked at whole brain data at all. I've really taken a zoomed in approach so that I can look specifically within the MTL. So if I'm really interested in knowing the difference, for example, between the dentate gyrus and adjacent CA fields, I probably don't want to do any normalization at all, or I would want to use a type of normalization where you can preserve the landmarks. So there's these types of normalizations where you can kind of put landmarks between different key areas that you want to make sure that you maintain alignment across. That's one thing that I would recommend. Or if you want to do whole brain, that's great, but I would recommend that you check very carefully after your normalization to see how the hippocampus is aligning. The hippocampus itself, even though there are these morphological differences across people, in general, I mean, you only have, you have basically, everybody has a head, a body, and a tail, and so you can at least make sure that those are aligning. When you get into the MTL cortex, especially perirhinal cortex, that's when things get very complicated, because you can imagine if you're using voxel-based registration, or if you're using, for example, surface-based registration, where the those sulci are not mapping onto one another, it can be very difficult. And that's why a lot of the work that I do it in native space, and I manually define or use automated methods, but very carefully check it. So I would say it's still possible to do whole brain and to do normalization using, for example, ants works very well. But just keep in mind that you're not going to have perfect alignment of some of those structures that are highly variable across subjects. Does that help?
Yeah, thank you.
Okay. Yeah.
Okay. Thank you.
Really cool talk. I'm curious about, in the last 10 or 20 years, there's been a huge explosion in cognitive neuroscience of people getting intracranial hippocampal recordings and even single unit electrodes. But one thing that seems funny about a lot of those papers, right, is they'll get these beautiful like single neuron or electrode LFP recordings from human hippocampus, and then it's sort of just reported as we have X number of neurons in hippocampus or MTL, and they're sort of all grouped or analyzed together. And so I'm curious, like, if your subfields group or others have tried to like coordinate with some of the people getting the electrode recordings to see if like you can maybe better demarcate like where they're recording from, or like use those protocols and those subjects or something.
Yeah, I think that one of the reasons why they don't always have the most precise localization is because, and this is a generalization, so there are some labs who do try to say that they're recording from a certain region versus another, but one of the problems I think is just the post-surgical MRI is subject to such bad distortions that you lack the same confidence that you would have if you didn't have an electrode implanted in somebody's brain. So I think that's one problem, but you can certainly be a little more specific in terms of lateral versus medial, or anterior versus posterior, head versus body or versus tail. That you certainly should have enough resolution to be able to tell. The other thing that they could do probably is, similar to what they do in animals, where they do like maybe cluster analysis on the different firing patterns. DG, for example, it's going to be really different than CA firing patterns. So that's another thing that they could do to try to really make it a little more clear. But one thing that's sort of interesting, though, is some of those studies actually find really similar patterns, for example, in the amygdala and the hippocampus, which is always really interesting because we think of the amygdala as doing something very different. But some of the UCLA studies, for example, find kind of similar firing patterns in those regions. But yeah, I agree. It would be nice. It's always a little dissatisfying when you just hear that it's hippocampus and you don't know where in the hippocampus it is. Yeah.
Okay. Last question.
Thanks very much. Really exciting talk and great resource for the community as well. So I was fascinated to hear about how much variability there is in the hippocampus. I didn't know that. And I'm curious whether there's any relationship with variability in cognition and behavior. Has any of these studies been done?
Yeah. So I'd say that traditionally people haven't paid out. There's always been labs that have tried to do shape analysis, but up until now, most people have just looked at volume. And certainly when you look at people with neurodegenerative disease, the smaller the volume, the worse the scores. But within the healthy individuals, healthy people, there's also quite a bit of variability. But that story has not been as clear. So within healthy young adults, the volume versus the cognitive function has not always been clear at all. Now I think there are some more surface-based methods that that are going to be allowing us to look a little bit more about those morphological differences. So for example, that dentation pattern. I know that there's some preliminary work from Ali Khan's lab that has looked at the relationship between those dentations and certain cognitive functions. So that's pretty cool, and hopefully there's more to come. In terms of for example, the tail, when it curves up versus curves down versus curves medial, I have no idea. So we'll have to do the research to find out.
Watch this space.
Yeah.
Thank you very much again for Rosanna. Yes.